Okay, here we are. We're going to try to draw a cell. And so I'm going to start with a sort of a generic circular type cell. And then here, make some little tiny finger-like extensions. Those are called microvilli. Ooh. Microvilli. Micro small, microvilli. And then usually the largest structure in the cell is going to be the nucleus. So here is the nucleus. The outer boundary for the nucleus is called the nuclear envelope. And the nuclear envelope has little circle holes all through it. Those are called nuclear pores. Okay, and then continuous with the nucleus is a flattened framework of membranes. Okay, so continuous with the nuclear envelope, you have a membrane-bound structure that's a bunch of flattened sacs of membranes. And then studded on the outside of this structure are ribosomes, which are involved in protein synthesis, making proteins. So if it is a flattened structure with ribosomes on the outside, what is that? That is going to be the rough endoplasmic reticulum. Okay, and it's rough because of the presence of those ribosomes. If you have flattened sacs that have no ribosomes on them, then it is called the smooth endoplasmic reticulum. Reticulum. There we go. Okie dokie. And then another structure that looks similar to this is the Golgi complex. The Golgi complex is also a big stack of membranes too, but it has a different function. It's involved in exocytosis, which means secreting things out of the cell. So it packages proteins and sometimes modifies them and gets them ready for export. And so this would be the Golgi apparatus. One of the things that's distinctive about the Golgi apparatus is these sort of fattened ends where they're kind of round and bulbous. And the reason for that is because those flattened ends will butt off and create vesicles. And those vesicles will then float up to the surface and they will fuse with the surface, and then they will secrete whatever was inside of them. So for example, if you have a hormone that you're trying to secrete from the cell, the hormones would be packaged inside of those vesicles, and then once it fuses it, then the hormones would be kind of released from the cell. Okay, so if you see a flattened sac with bulbous ends, often it's not located near the nucleus. That was going to be the Golgi complex. Golgi complex, also known as the Golgi apparatus. Whereas the rough endoplasmic reticulum, it'll have flattened ends, and it's going to be continuous with the nuclear envelope. Okay, so this is the Golgi complex, this is the rough endoplasmic reticulum, and then the smooth endoplasmic reticulum with no ribosomes. Okay, next is the mitochondria, and the mitochondria are distinctive because mitochondria are double membrane. Okay, so you have two phospholipid bilayers instead of one, and the inner membrane is going to create these folds. Those are called cristae, and that's going to help increase the surface area inside of there. And the reason for that is because the machinery that's involved in creating ATP, which is the form of energy for the cell, those are embedded in that inner membrane there. Okay, so this right here, this is a mitochondria. Mitochondrion, actually, because it's singular. If you have more than one, it would be mitochondria, which is the plural form. So mitochondrion, if it's one, mitochondria, if it's multiple ones. So you usually have a bunch of them. Okay, and they all have that kind of stripy picture inside, with the inner membrane in there. Okay, let's see, what else am I missing from the list? Plasma membrane. We never did the plasma membrane. The outermost boundary for the cell, that is going to be the plasma membrane. Okay, so this whole structure around the outside, that's the plasma membrane. And centrioles. For the centrioles, those are a little tricky to draw. They kind of look like crossed, sort of crossed fibers. Okay, and the centrioles, those are what create the cytoskeleton. So they kind of have microtubule fibers kind of coming out of them. So this would be the centriole. Okay, that's everything. Bye-bye.